Select the restriction in endonuclease enzymes whose restriction sites are present for tetracycline resistance gene in pBR322 cloning vector. So the answer will be option 1. Yes. So we did it in around 20 seconds, 15 to 20 seconds. So this was a diagram based question and without a diagram. So for preparing this topic, what you need to know is this structure. This is the structure of pBR322 plasmid. It is a cloning vector. Why? Because it has all the attributes of a cloning vector. So number 1, the origin of replication. It can replicate and make many copies within the cytoplasm. Then this part provides the enzymes required for replication of plasmid. ROP stands for replication of plasmid. Then it has two selectable markers, ampicillin resistance and tetracycline resistance. So the question which was asked here, it was asking for the two recognition sites that are in the region, in the open reading frame of tetracycline resistance gene. So BAM-H1 and SAL1. So in order to remember these names, BAM stands for Bacillus amyloliquefaciens and SAL is Streptomyces albus. This is just additional information. If you don't want to read it, don't, because don't overload. But in my case, I personally feel that I should know the names of these enzymes, because then it is very easy for me to abbreviate it and remember the abbreviations. So in this case, you won't get confused. But what about this? PVU and PST. Don't get confused between, no, don't make it PVT or PSU. See, PVU, Proteus vulgaris, and Providentia stewarti. And what else? You can see these restriction endonucleosides, they are not in within AMP and tetracycline. So if you use these restriction enzymes, it won't cause insertional inactivation of these two resistance genes. So EcoR1, ECO stands for Escherichia coli, CLAV1 stands for Caryophanon latum, and HIND stands for Haemophilus influenzae. The last one, PVU, again PVU stands for Proteus vulgaris. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8. 8 different sites, cloning sites are available in this plasmid. Now one thing which you should understand is what is insertional inactivation. Supposing if I use BAM-H1, what will happen to the tetracycline gene? It will get inactivated because this is the reading frame. So starting from here, the mRNA will be synthesized, okay, the transcript. If you insert any foreign DNA in between, what will happen? The open reading frame will get disturbed, and therefore the corresponding enzyme will not be synthesized. So this kind of loss of gene due to insertion of foreign DNA is called insertional inactivation. So in case someone uses BAM-H1 for insertion of foreign DNA, then the resultant transformant cell will be sensitive to tetracycline because the gene for tetracycline is now disrupted completely. But the transformant colonies will be resistant to ampicillin, and this is how we identify them. Their ability to resist ampicillin, but their sensitivity to tetracycline. It helps in identification of transformants. So let us revise once again. This is the cloning vector. This is the site for origin of replication, ORI. This is another important site for proteins involved in replication of plasmid, ROP. And which restriction enzyme the site is present here? It is Proteus vulgaris 2, PVU2. Then coming to the marker gene, selectable marker, this marker is AMP resistance, ampicillin resistance. And the two restriction sites are, first one is PVU, it is 1, and the other one is Providentia stewarti 1. Then over here is the tetracycline resistance gene, TET-R, and the two enzymes recognition sites are BAM-H1 and SAL, Streptomyces albus 1. And over here are EcoR1, Escherichia coli R1. Then here we have, let me make it like this. This will be Clavin, and last one will be Hind 3. So keep revising and redraw it, and this will, when you draw it your own self, you will realize that you can cross check yourself whether you know each and every name of restriction site and the markers and the location of ORI and ROP. So practice this diagram, and you will be able to attempt such questions very easily. That's all about this topic. Thank you so much for watching my video.